So now we've got differences between bacteria and archaeans. We talked about a bunch of different archaeans and some structures in archaeans. Now I want to talk about the different bacteria, different groups of bacteria. Again, we're looking at the major groups of bacteria. I'm not going to be terribly interested in you being able to regurgitate what goes into what group, but I do want you to see that there are differences. I get a lot of students who are just like, well, bacteria is a bacteria, and it just makes me sick. They are very different metabolically. They're very different. They look very different. They're as different from each other as you are to a fungus, heck, as you are to an amoeba. There are 30 phyla of bacteria, so subgroups of bacteria, and millions upon millions of species of them, most of which live in symbiosis with eukaryotes. What does it mean to be in symbiosis? Yeah, you're working together, you're sharing the space, you're mutual benefits. So bacteria usually aren't out to get you, contrary to what we just saw. If you were to look at life, you've got the eukaryotes over here, and then you've got a whole bunch of different groups of bacteria on the other side. The biggest distinction people tend to make with bacteria is the idea of gram-positive and gram-negative. You may have heard those terms before, gram-positive bacteria, gram-negative bacteria. This is a notation on how their phospholipid membranes are set up. Gram-positive will stain one color, gram-negative will stain another color. It's not that there are just two groups of bacteria, gram-positive, gram-negative. But gram-positive and gram-negative bacteria are going to react different ways to antibiotics, which is why it's so important. It's a very practical way of very quickly saying, this bacteria you can use antibiotics on, this bacteria you can't. There's a lot more to it than just two groups. You might have the chlamydiae. The chlamydiae are obligate intracellular pathogens. What does it mean to be obligate? Actually, it means need. I'm sorry, I didn't go over that yet in this course, I guess. It means they absolutely need. They're obligate, like obligation. Obligate intracellular parasites. That means pathogens. That means they have to have another cell to reproduce. They need to live inside of another cell. You may have heard of various types of chlamydiae. In chlamydia, it's a sexually transmitted disease. It enters into other cells and uses those other cells in order to reproduce themselves. And I love this. The green non-sulfur bacteria. Named such because they are green. And they do not live in sulfur-rich environments. If they're green, what do you think they're doing? Photosynthesis. Actinobacteria. This is a big group of bacteria. Their job is pretty much to break down carbon-containing molecules. These guys are going to thrive in the dirt. They produce a lot of secondary metabolites. So those are molecules that are important for, but not essential to, growth and reproduction. Planktomycetes. These are aquatic bacteria. Aquatic, of course, meaning they live in the water. And they reproduce by budding. So not through mitosis like you would expect, but little piece of it bud off and go off on their own. Typically, they have some form of compartmentalization. And the way they compartmentalize is through what's known as invagination of the membrane. That is, a piece of the membrane, if you've got a cell, the membrane is going to sort of invaginate in like that. And now, you've got two distinct compartments to the cell. Does that make sense? It's all still one cell. There's no internal compartmentalization. There's no organelles. But you see it's divided into regions, and different metabolic processes can happen in different regions. Spirochetes, they're chemoheterotrophic. What is chemoheterotroph? Where do they get their energy? Do they get it from the sun? No, not the sun. They're chemoheterotrophic, which means they're going to need to take in external chemicals, organic chemicals. Fusobacteria are obligate. What does it mean to be obligate? Necessary. Anaerobes. What's anaerobic? Absence of oxygen. They're obligate anaerobes. They're usually involved with human tissue necrosis. Necrotizing fasciitis. It's flesh-eating bacteria. Is a fusobacteria. It absolutely, in the presence of oxygen, it does not do well. So it kills the tissue around it, and when that tissue is killed, it does not transport oxygen to it. Cyanobacteria, named such because they are cyan in color. Cyanobacteria, this greenish color, which means photosynthesis, because whenever you see green, think photosynthesis. Cyanobacteria are insanely important to life on the world as we know it. Before the cyanobacteria, there was no oxygen. Once they came into play, they started taking in this carbon dioxide that was readily available in the atmosphere and turning it into oxygen. And that oxygen is super highly electronegative, which means it steals electrons. It oxidizes things. Before you had cyanobacteria present, there was no free oxygen to break other things down. Thermophilic sulfate reducers. All right, thermophilic, what does that mean? They like heat, and they are sulfate reducers. By reduce, it means to do what? Right. So they're going to gain they reduce the sulfate. That means that they're going to add electrons to sulfate. They reduce the charge of the sulfate. Acidobacteria. These are fairly newly discovered. Like, we found a whole new group of bacteria. And they're present in soils, and they're very likely important for recycling carbon. So carbon is going to be released to the atmosphere as carbon dioxide or fixed as sugar or some type of carbohydrate. So these bacteria are important for converting the carbohydrate back into CO2. Proteobacteria, they're gram-negative, but they are they're involved in nitrogen fixation. Nitrogen fixation is insanely important to life because you need nitrogen in order to build amino acids. These nitrogen fixers take N2 out of the atmosphere and form it into ammonia. And that ammonia is then used to build amino acids. We cannot do that. Our metabolism does not allow us to. Plant metabolism doesn't allow it to. It's the bacteria that plants rely on that allows this to happen. My favorite strain of bacteria, because I do have a favorite, is Agrobacterium tumefaciens, which is going to work as a it typically works as a nitrogen fixer. But what it does is it changes the DNA of plants. Literally, this bacteria will infect the plant. And where it infects the plant, it inserts the bacteria's DNA into the plant, forcing the plant to grow these tumor-like structures that house the bacteria. Sometimes it makes what are known as hair roots grow. They are natural genetic engineers. The genes of the bacteria are inserted into the genes of the plant stably. We, as humans, have a super hard time doing that. We cannot, using our current level of technology, genetically engineer a plant as well as a bacteria can. And that's amazing to me. We can, though, and this is where we get smart and we get credit, we can genetically modify the bacteria. And if we genetically modify the bacteria, and then the bacteria does all the work in modifying the plant, we're like, we totally modified the plant. It was the bacteria that did it. It's using these agrobacterium that we're able to create all sorts of new plant products. So all because this bacteria is better at genetics than we are. Bacteria, and archaeans to an extent, all have this issue with what's known as horizontal gene transfer. This is where taxonomy sort of falls apart. Because we've defined in this class a species as a group of organisms that can interbreed with each other. But what happens with horizontal gene transfer is two bacteria come together of different species. They send a pilus across, a little hair. They touch each other. And DNA can travel from one to the other. That's a conjunction. You can have a direct transfer of DNA. Heck, bacteria are super special because they can just be wandering around, find some DNA in the environment. Not kidding. Literally, one's like some spare DNA. They take it into themselves, and it integrates into their genome. And now they can express that. They might get a virus coming along. It's called transduction. A virus comes along, passes its DNA into the bacteria, and suddenly the bacteria is expressing the viral DNA. Bacteria are not terribly picky about expressing DNA as long as it's present in their cells. You guys, me too, I guess, our cells, if we pick up foreign DNA, we're like, that's bad DNA, we kill it. Bacteria pick it up and they're like, might use this sometime. Oh, it's sort of like you guys keep random cords at your house that go to something and you don't know what it's to, but by God it's to something and maybe it'll come in useful later. That's what bacteria do with DNA. And then they start just randomly plugging it into stuff to see if it happens to work. Sometimes it works, sometimes it doesn't. Sometimes the bacteria gets an advantage, sometimes it gets a disadvantage. When it gets a disadvantage, what happens to the bacteria? It dies. What happens when it gets an advantage? No, it doesn't die. It breeds faster, and suddenly it starts outcompeting other things. And that's evolution. This horizontal gene transfer, just picking up random bits of DNA, increases genetic diversity. We have, up till now, looked at phylogenetic trees as fairly straight lines. But the truth is, there's all sorts of connections between the bacterial lineages. You're seeing lots and lots of, instead of just straight up lines, lots of horizontal lines connecting branches. Probably the one that you're going to be most familiar with is think about plants. Plants and, never mind, let's think about animals. Eukaryotic cells, right? Eukaryotic cells took in, at some point, a brown algae that became its mitochondria. Do you guys remember what that's called, what that theory is? Endosymbiosis, exactly right. Endosymbiosis theory. So with endosymbiosis, you've got this bacteria that has its own set of genes in with another organism with its own set of genes. They come around and pick up an entirely different species, a cyanobacteria, take it in. All right, well, now we have another species with its own genes. That one cell has effectively 3 distinct genomes, leading to this transfer back and forth. It gets confusing.